as you can see, she's the director of the Eye for Healthcare and Life Sciences at the IBM Research in Haifa. She's also professor in computational medicine at the Hebrew University in Jerusalem. And she's starting two very interesting activities for which we will hear more, I guess, these 2 days. One is the Alliance for Drug Discovery, that is very much related with things we are interested, and the other one is the International Network for Life Sciences. That is also very much related with things we are interested. And going through her papers, she has done, just choosing keywords of things that are very much related with things we are interested. Obviously, the work with real-world data, the application of network type of approaches, network science to biological problems, all the now multi-modal combinations, electronic records, imaging, and all that. That is also obviously very much interesting. The emphasis on explainability, how this is going to apply to biomedicine and this explainability. She has also worked in disease trajectories. That is also something that we are very interested in we are doing too. And I find a paper from 2017. She was discussing how to integrate artificial intelligence in mental health and justice. That is, now this is long ago. 2017 looks like a century ago. It's not that far, but it looks long. Now this has become a very hot topic, because now we know more about the pros and the complications of the integration of artificial intelligence methods that are now pervasive. So now, with all these many similar keywords, we just look forward.
Thank you very much. Thanks. I'm very happy to be here. And this is my clicker, right? Good morning. Thanks a lot for the invitation. I'm very happy to be here, and I'm going to talk about the multimodal foundation models for drug discovery. Thanks for reminding me on other things that we did, that we enjoyed a lot doing in different formats and ways, and I'm happy to talk about them, but at a different, some of them are not part of this, like the mental health. It's a whole open issue that we can discuss. We all probably divide the world of AI into pre-gen AI and generative AI. There's this, in my, everyone has his own history and perspective. So from my perspective, when I came back from a postdoc at UC Berkeley to Israel, it was 2005, and I was doing AI, but AI was try whatever architecture, method, approach. It was Bayesian networks, it was support vector machines. It was very, very wide in terms of what methods you are using, but very narrow in terms of how much data you can access to and analyze, right? And then there was the whole change into deep learning, and that was more or less from that time, 2010 to 2020, where with all the competitions, it was obvious that deep learning offers the most accurate results, all the competitions on large data sets. And the data increased accordingly as well, and there was ImageNet from Google and so on. And then in 2020, a team in Stanford coined the term foundation models and talked about, well, it's not only deep learning, but there's this approach of large language models that you are all expert in, where you have a pre-trained model that benefits from asking, and then after you have the pre-trained model benefiting from huge amount of data, then you can fine-tune it and perform inference on smaller data sets. And that's the era of generative AI and foundation models. And everything that I'm going to talk about today is from this era. So everything I did before that, long done in history, and we are looking at everything from the perspective of what can we achieve with these much newer and stronger technologies, obviously. And I'm going to share with you today what we are doing at IBM. And this is focusing on the very diverse set of information that can be accumulated in biology from omics data, genomics, epigenomics, and so on, and trying to infer from that basic biology that will support the discovery of drugs. So we are looking at the very initial stages of drug discovery, from target identification to target validation, which typically, it's a protein that you look for that has a significant role in the process of evolvement of a disease and symptoms of a disease. And once you identify this protein, that if you manipulate it in some way, potentially with a small molecule or a biologic that will interact with it, then the disease course will change, and potentially you can get a full cure or reduce of symptoms or stop of neurodegenerative disease where you stop this process. So in all of these stages, it seems that if we better understand the cells and the interactions between molecules and our cells, we can offer much stronger suggestions of what would be a target and how to optimize it, and what would be a lead, a molecule, and how to optimize it. That's what we are doing, and we are supporting discovery. As you know, this is an interesting paper that talks about how AI already changes today drug discovery. So in this paper, you can see, and this is from last year. I trust that if someone would have done the same exercise already in January 2025, more information will come to play, because here they report on a single molecule that is already launched that is based on AI, and many others that are in the process. I must say that I look at this with a grain of salt, right? It's at the end, many teams are reporting of using AI, and not necessarily it's the full complete AI. There's a full hype around it. And yet, it's very interesting to see these numbers. So that's why I shared it as a prep for what I'm going to share today. What we've created at IBM is biomedical foundation models, which is a family of models that addresses these unique data sets as suggested. And after we create these models, we want to support, as I said, discovery of targets and so on, targets, small molecules, and biologics. And obviously, a key element is validation, how we validate what we are doing. So we have different processes for validation. I will come from the weakest validation method to the strongest one. So the weakest would be alternative in silico methods. We have one in silico method. We take a different prediction models, and we see if we have agreement between them. In the case of protein-protein, if we want to design an antibody, so in the case that we look at protein-protein interaction, antigen and antibody, we want to design an antibody given an antigen. So the antigen is our target. We want to design the antibody. And we want to create an antibody that first and foremost binds to the antigen. So we use our in silico method, and we designed a new antibody, and we predicted its binding energy. A very weak validation would be we go to any of these existing AlphaFold-like models that predicts structure, and they predict structure of a complex of proteins. So they also predict structure of the antibody and the antigen interacting together as a complex. And when they predict this structure, they give some confidence of how likely this is a stable structure. So this is taking from a totally different angle a validation to what we did. But this is still weak validation, obviously. Then there is another weak validation, but somewhat stronger, which says, oh, it's not about agnostic AI tools. Let's go to the molecular dynamic simulations that we can do for these proteins. If in the concrete example that I just provided, where we have an antigen, an antibody, and we want to predict that they bind or not, or do not bind. And of course, we benefited from generative AI, so we created this, we designed this new antibody. Maybe it's a template-based design. So we had a known antibody that binds to the antigen, and we created mutations. We changed some of the amino acids in the antibody in order to create a new one that we expect it also to bind to the antigen. In such an example, we can now put it in a molecular dynamic simulation tool and see if, according to the physics of these molecules, it makes sense that they bind in the epitope at the place we expect it to bind. This is also somewhat weak because you might, I don't know how many of you have tried to use molecular dynamic simulations to validate anything you're doing? Can you raise your hand? Just so that I know if very few, okay. Because it also has its limitations, right? Because the molecular dynamic simulations first often it's just in the void. It's like two molecules trying to interact, and in reality it's a different thing. Okay, but the algorithm is doing the same. But it's not as strong as a validation in vitro testing, where you can test it in a medium that represents the cell and the intracellular materials. Second, you need to know the full structure and the pose of the molecules when they bind, when you want to simulate them. And if you just created a new protein and you design a new antibody, it's not necessarily that you can accurately predict its structure in order to initiate these simulations. So these are also limited methods, and of course, the next more strong evidence would be in vitro testing, and the ultimate goal would be test it in live people, and then you know if it does what you expected it to do. But to know if you correctly predicted binding, in vitro testing probably would give you that answer. And we work, as you can see, with a long list of partners in order to generate results. We signed the deal with Boehringer Ingelheim. We work with them on antibody design. We work with Cleveland Clinic on many different nanobodies, antigens, design prediction of properties for vaccines with small molecules generation. All of that, the whole full spectrum questions about targets. So these are questions about perturbation of cells. So if you knock out or knock down a gene in a cell and you want to predict what will happen, these are also these type of questions our tools also can offer answers, and we do that with the universities and as well as with Cleveland Clinic. And as mentioned, IBM and Meta created an AI alliance at the end of 2023. And BSC is part of the AI Alliance. IBM is part of the AI Alliance. There are many, many partners, more than 100 partners in the AI Alliance. And what we created recently is an AI Alliance working group on AI for drug discovery. And this working group was kicked off in New York, in Boston, in last 3 months ago, at the end of October 2024, and it was well attended. And we created 2 working groups. One is working group on multimodality, and another working group is about validation. I'm sharing this here because it's an open invitation. BSC is already part of it, so if you are part of BSC, it's automatically you can join whatever activity you are interested in. And if you're interested, please do feel free to reach out to me. I'm here the next 2 days. I would love to talk with you. I'm co-leading the working group of AI for drug discovery, and specifically, I have strong interest in the multimodality. So I'm co-leading with a professor Professor Alex Shellac from MIT and Professor Nir Friedman from the Hebrew University, a multimodality we call it a multimodality project. And I would love to have others to join us. So do feel free to join us. So this is about the AI for drug discovery. Working group in AI Alliance. You did mention that we are also I'm a director in a new organization for AI for life sciences. And there, it's the opportunity to support each other in the academia. So this is with my academic hat. The Hebrew University is also part of the AI Alliance, but now with my academic hat, if there are advisors of PhD students who want to share knowledge and to test each other's students and so on, so we created this other activity network that I shared in my LinkedIn, so you can see more information there. And again, feel free to reach out and to ask me questions. I just realized that I didn't look at the time, and I need to be on top of time. So, but give me a sign like 5 minutes before. Okay, perfect. So this is just as a reminder to all of us how complex diseases are as a motivation to the technology that we have developed. So this is a paper we wrote in 2020 with a number of colleagues from Switzerland about the complexity of cancer and how cancer as a disease is heterogeneous in each and every aspect. So from person to person, within the person over time, as well as within the person in different places, what would be called the same cancer disease would have different cells. And that means that the disease has different faces and can act in different ways. So if we, from the data and AI perspective, want to support physicians in better understand the disease and offer cure for such diseases, we need to be able to analyze these complex data sets and to be aware of how heterogeneous they are. Similarly, we did this paper I wrote with Professor Fei-Sheng Cheng from the Cleveland Clinic, and this is about the opportunity of AI in Alzheimer's disease. And it repeats the same story, but in a different disease. Obviously, each disease has its own most important data sources to learn about the disease. So if in cancer, a key data source would be the cell analysis and the pathology, which includes the pathology and the imaging, as well as, of course, other elements. In the context of Alzheimer, it is much more complex, because to understand what's the status of the Alzheimer disease, you might want to track the language that the people are using in different ways to understand the dynamics of the disease and learn from it. While at some point in the past we were addressing many different facets of patients, we are now very focusing on the core biology, so on the questions at the microscopic level, and we provide answers to these questions. But as you will see, we created a technology in a way that enables orchestration and interaction with models of other teams. So if a team learned about imaging of pathology, and we are learning about transcriptomic of the cells, and we want to be able to offer support to understand the disease from both directions, then we have a way to orchestrate it. What I'm going to share today are two approaches that led to two specific models. So there's the melon approach and the mammal approach. I will elaborate on both of them that we created. In each of these approaches, we offer an open source code in Hugging Face and GitHub repository. So I will share that you can look at it and evaluate yourself. And it addresses this challenge that we have. We understand that when we want to learn about these physical entities, proteins and small molecules, and activity of cell, the representations or the views that might be important go beyond the sequence. So when you look at small molecules, often people are looking at the SMILES representation, which is a standard way to convert a three-dimensional molecule into the world of one-dimensional sequence. We tested the potential of integrating different representations. So we looked at sequence, image, 2D graph, and 3D structure. Although we believe that there's also the fourth dimension, of course. So when you think about activity of a molecule, it also changes over time. And in some cases, in the example I gave before about cancer, as you know, a lot of the new therapeutics are offered based on a unique activity of the cell, where we want the immune system to identify a cell as cancerous. So we can ask questions about the immunogenicity of the cell, how likely that we can identify it as cancerous. And the way for the immune system to identify a cell as cancerous is by examining the peptides, the remaining of the proteins that are in the cell that are displayed on HLA. So this display on HLA, it's a full dynamic story, right? There's the HLA capturing the peptide in the cell, moving it outside, then the antibody from the immune system might come and wants to inspect whether, sniff it, some people call it, to inspect whether this is a cancerous and identify that as cancer, and this is an immunogenicity property of this HLA and cell or not. So we have a different study with molecular dynamics and deep learning and so on, where we analyzed 4D, the three-dimensional, as well as the dynamic over time. But to simplify things, we are not yet in the 4D full steam. We suggested an approach that the current model only limits to sequence, image, and 2D graph, the model that is outside in the world. We also do 3D. And similarly, in mammal approach, we have small molecules, protein, RNA, and DNA, and free text we are doing internally and we'll share at some point. Just to be on the same page, we talked about generative AI and foundation models and the fact that you use masking. So there are many different approaches to masking. It repeats itself when you look at DNA, when you look at RNA, drug molecules, protein sequences. There are many different ways to create tasks, self-supervised tasks to the model to make sure the model captures well what the biology means, what are the interactions in the biology, or what are the associations in the biology, in the biological context that we expose to the system. And we use strategies that go far beyond this, and probably this is not needed for this audience, but just to make sure we all talk at the same language, I give some examples of how we do self-supervised task learning before before we use it for fine-tuned tasks. And this is, as I said, there's interest in 4-dimensional, and we look at all of them. What we share externally, it's not yet the full-fledged story. So I share that I'm going to talk about melon and mammal. I'm starting with melon, which is small molecules going all the way from sequence to 2D, and we have now an internal version 4D. This is in arXiv, and it's available externally. And I guess you're fully aware with the different ways to combine modalities in language models. Very, very simplistic way to put the options is either you do early fusion or late fusion, right? Either you already enter the data from the very beginning and the representations from the very beginning and you train the model, or you have different models trained separately and at the very late stage, you combine them, you do an ensemble. So that's the late fusion. And there are strategies on how to do late fusion and strategies on how to do early fusion. Our melon approach is based on a late fusion approach. So we have a model for 1D, a model for 2D graph, a model for 2D images. And in late fusion, we have multi-layer perceptron that combines all of these models to offer a strong result that benefits from all representations of the molecules. So that's late fusion approach, and that's a key element of the melon approach. And it uses data, 200 million molecules from PubChem and Zinc 2022. We compared and contrast with other models. There's Molformer, there are many other models of small molecules, and we saw that it's not always important to have the largest data set. It's more important of small molecules. It's more important to have druggable-like molecules that you learn from in order to be able to provide properties and generate new small molecules that are candidate for drugs. And there are different rules that tells when a small molecule can be a drug. And we took data from this zinc that has billions of data points, a subset that we believe, some of it randomly sampled, some of it is cured and specifically selected in order to fit drugs that small molecules that represent drugs. And you can see that we compare our model with GCN is the baseline, MultiView is our melon approach, and we have the separate modalities listed as well. We used 21 tasks for MoleculeNet, and the higher the better in this radar plot. And you can see that when we compare, almost always it's better to combine the models. So to have the late fusion approach of multimodality for small molecules, when you ask questions like different properties, each one is different fine-tuning task and predicting the property, and you want to get closer to one, and you can see that the model offer very strong results. And this is an explanation of how we perform the late fusion. So as I said, it's a multi-layer perceptron that has different weights, that provides different weights, learns the different weights. Alphas here are the different weights that each of the modality is assigned with. And you can see here some analysis of what are the weights in the different tasks. And you can see that the value of the weights represents in some way the importance of the modality. The higher the weight, the more important the modality. It's not exactly, but I mean, it gives some sense of the importance of the modality. And what we see here is that the modalities, graph is an important modality, but it changes. Sometimes the sequence gives a very strong, sometimes the text, which is sequence, is assigned with a strong weight, sometimes with a very low weight, but all of them contribute. There's very few zeros. There are, but very few zeros where a certain model was not important at all for that particular task. So that's what you see here, that the combination makes sense. That was, up until now, was melon. I'm stopping here for any comment or question or suggestion, or if you prefer, we can do it at the end, whatever way you prefer.
interesting. The different modalities are contributing in a very different way, even for the cytochromes that should not be that different.
That they change their weights, although it should be similar. Yeah, so that's why I said we need to be a bit careful with the interpretation. So the one with 1 and 0s is a bit weird to me. But at the end, there's some permutation. So we don't know if the alternative of 2 were, I mean, similar information comes from the different modalities. So sometimes one gets higher and the other gets lower, but it might not be that significant in terms of the importance. That's the reason for all the disclaimers I gave. And thank you for the feedback. Yeah.
So there are some enzymes here and some tox21, toxcast. What was the prediction there, or what was done there? Because there are multiple endpoints now in tox21, so I don't really understand the...
So I need to go back. There is a paper, you can read it. Tox21 is 12 different toxicity measures, and I don't remember if they used all 12 or just a subset. I need to go back to the paper.
Okay, because it's interesting to have just one number
for each one of the 12. Yeah. First, it's an open source code. You can test it yourself, see what results it gives, and so on. And it's in the paper. I don't. Okay. I mean, I don't recall all of these. And thanks. I think it's. I see someone online. Patrick raised his hand.
Yes, thank you very much. So now I'm just. Can you hear me?
Yes, very well.
Okay, very good. So I'm surprised to see that. I mean, MoleculeNet has a different level of complexity task, right? So you have things here like the binding to the beta secretase or penetration of the blood-brain barrier or to the HIV protease. That these are kind of tasks that only rely on the chemical structure of the components and a little bit on the target, right? But then you also have tox21, toxcast, and all these things that are a lot more complex. But at least according to this slide, I mean, it seems that for these more complex tasks, your perceptron is mostly using only graph information without leveraging anything from image or text. While in the easier task, and that I'm sure that all the predictors, including your own, do better, it leverages different types of information. Have you considered this?
It's a good point, and let me just go back to the results. Although the measures are not always the same measure, but you can see that tox21 performance was not as good as the other ones. So it aligns with your thought of it's a more complex one. I will have to think further about that, because given that it's a complex one, maybe the text doesn't capture much about the very complex tasks that we have there, and that's why the graph is a better representation. I don't know why the image does not contribute much, but we will have to test
Thank
that.
you.
Yeah, that's a good point. There's, we came to this challenge with analyzing all of these tasks, and we focused on one specific question, which is a target we cared about. So part of what we do in the AI Alliance, and it's not us, there's a group of people who met in Boston. Part of the discussion was that the benchmarks are very limited and not good in capturing the properties that we expect. And it seems there's a need to generate better benchmarks to begin with. And because of that, we give low weight in our mind to many of the things that are happening in the benchmarks, and much higher weight to the molecules we cared about that we are now testing in vitro. So that's why I'm very cautious in commenting anything on what we learned from these type of results. We just learned that the quality of what we achieved is at the same level of competitors, and it's capturing what foundation models are able to capture today. But I wouldn't give much attention to each and specific value, because there's a lot of debate on the how good these benchmarks are, and we are working on creating better benchmarks. I think the same applies to the second part. So the second part is mammal, and I'm going to talk about biologics challenges. Mammal is an approach that addresses multimodality, and in melon we only looked at small molecules. In mammal we also look at biologics, at proteins, and as you know that open up the scope of industrial and important question we can answer, because now we can design new nanobodies and antibodies and predict binding affinity and do all kind of predictions on other properties and design vaccines. And these are the type of questions that we are addressing now with mammal, along with other questions. Again, there's a paper out there. The key aspect of mammal is that it's a very flexible language model, sequence-based model, essentially, not language, sequence-based model that enables as input different domains. You can provide as input the small molecule and the biologic that interacts with it, or the sequence, the expression levels of the messenger RNA in the cell, the transcriptomic data, as well as some specific biologics, proteins, and some specific small molecules. So all of that can be represented well, and it is very flexible, so it can provide answers on very different tasks and very different questions. And it's based on the architecture of T5. We are now testing also alternative architectures, but what we offered was T5-based architecture. It has multitask pre-training approach, so we not only limit ourselves to the masking that I showed before, but to many different complex strategies of how we pre-train. And then it offers answers on many different domains. I think I said all of that. It's encoder-decoder and encoder-only algorithm, so maybe that's the only thing I didn't highlight here. It can get as input numeric tokens, not only categorical or word-like tokens. And in this case, we took data from many different sources in order to do the pre-training. So we pre-trained on 2 billion. Beforehand in Melon, we talked on 200 million. Here it's 2 billion data points that we use for pre-training, and you have the full list of what was the data and what were the tasks that we were using. It's from the paper. And the databases, so as you saw before, it was the same graph, but showing what was done in Melon. Now it's the same data of Melon, so the 200 million small molecules that we curated and prepared for Melon was used also in mammal. And we also used Select gene, so it's transcriptomic data and proteins data, like OAS and string and so on. So we used all of these data sets as well. And again, with the disclaimer that the benchmarks are not so great also in this domain, and we put much more emphasis on testing the specific molecules we design and predicting and testing properties that we predict. So we do have lab tests that we are performing internally with all kinds of partners, and as soon as we publish, we can share more about our internal testing. What we did here is the same approach. The SOTA in each of these tasks is different, coming from different source, because there's no one team who performed all of these tests. And what you can see here are different. It's a rudder plot of different tests, where if we did protein-protein interaction prediction of the energy, delta G, we learned only from protein, so it was a protein-protein. But when we wanted to learn about drug-target interaction, we learned from proteins and small molecules as input for the Melon model. Or if we wanted to do predictions on cancer cell lines and questions about perturbation studies with small molecules, so you ask about IC50, how much molecules you need to put in the cell in order for half of the cells to die. So this prediction was done where the input was the gene expression of the cells, as well as the representation of the small molecule that was used in order to perturb that cell. So with this approach, we have very high flexibility on representing different data types. And this can lead to many different questions. So what you have here are ways to reach out, and you can see the names of our biomedical small molecules and our biomedical omics, biologic small molecules, multi-alignment T5 base encoder decoder model. So you can see here, you can get from here both the Hugging Face models, the GitHub repositories, all of that is available for discussion. I'm now moving to the questions of how we evaluate these tools and what's the meaning of explainability. So I refer to one of the papers you mentioned about our explainability. But before moving there, how much more time do I have? Okay, great. And any question or any comment before I move to the last part of the talk?
Can you tell us more about, I'm sure you're trying antibody design and antibody binding, and so how many you think, so now we see that the top #1 company, Apsys, is doing thousands, tens of thousands of antibodies, and then they developed a language model that, and they can do this because they have ultra-fast experimental and very large range. And then, so we are working in antibody designs, but how many you think we need to do experimental validation on? I mean, on my take, today it's impossible to design 10 antibodies and get a binder one. And whoever says that is either biasing the data to one case or lying, because a single mutation will change completely the CD loops, and then you're screwed. So what's your take from your experimental validation if you're running on this? If you can tell us something.
Well, that's a very good question. We are addressing this question all the time, and there are papers that are very misleading, right? Sometimes they say, yeah, we found this new antibody, but then you look and they did 10,000 different antibodies, and then you say, okay, with this, yeah. Yeah, but then if you find 2 out of 10,000, I don't know if it's so cost effective unless you have a method to. So we are aiming at better hit than 0.02, right? Percentage. We are struggling with that, and the more we do, the more we learn about the noisiness of the lab tests themselves. Because often, without referring to the specific things we do, for obvious reasons, but often, if you do high throughput, then it's not accurate enough, and then you learn from this data, and then you want to get to the more accurate ones. So it creates a whole bunch of ripple effect that you started in a place that is not accurate enough, but you have more data, but then it can lead you to the wrong direction. So we are still mapping out the different options to understand what is the most cost-effective one. So if a high throughput screening gives us much less accuracy, but much larger data, and vice versa, a smaller setting gives you much better accuracy, but much smaller data. We are still considering and exploring. I can't really refer to a specific answer. This is the recipe for success, but we are working on that.
Would you say, last question,
yeah.
Would you say that today we need to have this wide window of antibodies and diversity plus experimental validation in order to create a dialect that will predict the next antibody for this particular target? Or you would say, no, I can do AlphaFold without evolution that will create me 1,000 antibodies in a minute, and then I can do some quick molecular modeling validation. Would you say that theoretical is ready yet?
No, I still think that we are data hungry. We are at this range, that data is insufficient, and to have stronger predictions, we need more data in this space. It's just that this space is very not homogeneous, because if you look at certain targets that are of interest for the industry, there's almost nothing there. So they don't want to share the target, but you don't have data there, but then you want to create data on the specific epitope, on the specific target that is of interest for that company. So it's not necessarily that if we get 10,000 other antibodies from some other place, we will benefit much. So we are still, because the whole space of what matters for us is not homogeneous at all. And so this is one element that we are definitely data hungry, and we are at a space where we need more data. But where and which and what, it's very complex. No, it's not like that. It's very complex. There's also this problem that we know very well, that we often only have positives. So it's not like that all of these data sets that collect from publications, publications, you don't share that you tried this and that and that and that, and it didn't work, and you know for sure it doesn't bind. So this is another problem that we have, the imbalance data that we need to face when we want to fine-tune our models and be more accurate. So yeah, this is a very challenging landscape and a lot of opportunity. We see, as you say, we see a lot of work on that space. Any other common question before I move? Yes.
So my question is mostly related with the validation process. So I've been looking at these spider graphs, and you seem to reach quite high on all these benchmarks. So my question is there a threshold in these benchmarks that you need to cross, that you are always trying to achieve? Of course, the higher the better, and all the caveats when it comes to benchmarking. But if you're going to deploy a model, if you're going to release the model in such a critical scenario, because I'm thinking, I work with text language models, and that is not a problem if your model summarizes and scores an 85%, right? But if you're using a model for something more critical, maybe an 85% is not enough. Is there a threshold that you try to meet or overpass before deploying anything?
That's a very good point, and actually it helps me move, pushing towards the last part of the talk, which is about validation and confidence and what is important in this world. So, as you said, these benchmarks are what the academic community uses in order to compare and contrast. It's not what the industry would use in order to decide, oh, I'm going to deploy a system. The industry will use on the specific targets they care about, on comparing to their own way to assess. At the end, it's a business question. So always you can do more lab tests and make the AI not necessary, but it's a question of return on investment. To design a complete new antibody, it's almost impossible for the industry to do with current methods, and generative AI should be able to do that. But if generative AI is so uncertain and you need to test so many times, you might just blank run as many as you can. So these questions are being answered in the context of the specific target and in the context of the specific question. And the community competed on all of these benchmarks. So what we show here in red is the best solution we found in the papers that also offer information about how they divided to test and train. Because sometimes you get a report of a very good result, but you cannot reproduce it because they used whatever way to design the test. And there's a lot of hidden details that are important how you design the test. So if you, for instance, this cancer drug, this pink part of the cell lines, this is taken from TDC, Therapeutic Data Commons, and there are certain papers there that share how they divided the drugs. But if you take drugs with the same pathways, they will probably lead to the same IC50. So if you design the test and the train in a way that in the test there are drugs you never saw before, but these drugs are very similar to drugs in the train, it's a very easy task and you'll get different results. But if these drugs are very different, it's much harder task, and the performance will not be that perfect. So it's very important to understand how you design the test, and for the industry to deploy, it's key. They have their own tests. So that's why there's a huge gap between what you see performance on the academic measures and what you see in practice.
Thank you.
And that leads me to this different question that we ask ourselves, a team of people from Europe, on the question was, what would be the best, we asked question, it's a different type of question, right? So bear with me, it's a different perspective. We asked, what's the community is doing. It's not like what's the right answer. It's like, what would the community of researchers working in the area of healthcare and life sciences would say about the importance of explainability? And to probe what the community is saying, we actually took PubMed and downloaded automatically all the papers that talks about explainable AI in biomedical. And then we manually labeled all of these papers, and we said, okay, this is explainable AI in biomedical. Are these papers about evaluation methods, new methods, discoveries, reviews, or tools? So what type of things are being developed by the community? And how many such papers are there? And how can we organize the information about what's explainability in AI is? So the answers we obtained based on our analysis was, first, explainability in AI can be divided into 3 different methods, very clearly, based on all of these papers. It's either causal, which is the highest level of explainability. You can learn from data the cause and effect. It's very hard, it's almost never happened, but that's like the strongest technology in explainable AI to infer causal relations. And we, as a team at IBM, have some tools in the open that we suggest how to infer causal relations. We worked with that. It's really very hard. But this is one method of how to explain what you obtained, because you found a new causal relation, and that explains why you can predict something or you can discover something. That's one method. The other method is embedded method, which is the AI by itself is understandable. The best example for understandable AI, can you imagine, from all of these methods that you can take from Bayesian network, support vector machine, deep learning, decision trees, what would be the most explainable technology? I think decision trees would be something that if physicians are being said, use this algorithm to predict, to get a prediction on how likely this person will get back to emergency room when you are now releasing the person. And you have a prediction. If the physician wants to understand this prediction and you give the physician a deep learning algorithm, what can the physician do with that? Or you give it support vector machine. But if you give a decision support, then it's very clear, right? Decision support system, you can say, decision support tree, you can say, first, the question is how old the person. If it's older than 80, it's likely. Lower than 80, it's much less likely. Then you go and you have a set of questions, and now the physician can track it and understand it. So decision trees seems to be an AI algorithm that it's embedded, the understanding is embedded in the way the AI algorithm is designed. But vast majority of algorithms we use today, it's not relevant, right? I do remember when I was like 15 years old younger, so 15 years younger, like there were conferences of healthcare and life sciences, and I met a lot of physicians who were talking about AI, how exciting it is. And some of them were like preachers. They were like preaching and saying, use decision trees. We want to use your tools, but we want to understand them. So please don't go wild and try all kinds of methods. Use decision trees. But it's no longer relevant, right? We know we have much stronger algorithm. We talked today about foundation models. So why would we use decision trees if it's not as strong as others? So the other method would be post-analysis, retrospectively to analyze the models. Am I at the end of the, I don't want to hold people, Simona, am I at the very end of my talk? Okay, yeah, so I'm at the very end of my talk. So I'll finish in 2 minutes to make sure that people can just come up and go and feel comfortable. So I talked about 3 methods: causality, embedded explanation, and post-analysis. Post-analysis, SHAP analysis. How many of you heard about SHAP analysis? It's a very common method, and it's based on Shapley's Nobel Prize laureate approach that says, how do I assign contribution to different players? It's a whole different story, but it's retrospectively you ask which one contributed to my answer, which feature, which group of features. And the SHAP is very in principle approach, but there are other approaches, and there's based on SHAP other modifications. So we saw that there was a big jump in interest in these things, much more than the jump in general in AI that was also a big hype. And we saw, I don't know if you know Eric Topol, a physician who is working on innovation. He tweeted about this paper because he really liked what we came up with that showed that COVID seems to accelerate all of this interest. So with COVID, physicians and experts were using more AI, but they wanted to understand it more. So there was much more a write-up about that. We have tools to understand bin benchmarks and so on. I'm going to switch to finish this and to just finish with my key messages. So foundation models transform drug discovery. Two novel approaches that I share with you are mammal and melon. And it is needed to design, it's very important to design strategies for assessing such models. And the whole discussion about what's explainability is a prep to what we would like to do also as part of AHEAD, is to be able to evaluate and learn and test how these models are being performed to make sure that they are not biased and to offer them in practice. This is open. And I'm urging you, saying, feel free to get into the open source tools and the Hugging Face models and so on. It's all in the open. And as part of AHEAD, we will also collaborate with people on how we evaluate these tools and create evaluation tools that hopefully are relying on methods that we already started to do that, that are relying on models that we already started to do that are up in the open as well in our GitHub, and you can see that. Thank you very much.
Thank you for the talk, fantastic talk. And question is about graphs, because if I got correctly, these foundational models are about LLMs, right? Multi-modal, but anyway, it's transformer based. And you also mentioned that you face problem with amount of data. If we have not a lot of data, probably we have to introduce some biases into model or some. And what do you think about using graphs? How we can use them if we can in this context? Thank you.
Graph to capture better connection between the data that we have to capture information about the data? In what way you are asking about graphs? Graph representation?
Yes, indeed. I'm asking about graph foundational models because they just started to appear, but I don't know how practical they are at the moment.
Oh, graphical. So graph neural network is out there for more than 10 years, and the foundation models version is something that we explore with. There are two ways to use the graphs. One is to map it into a sequence. So you have a graph representation, then you map it into a sequence, then it's easy to use. And the other one is to more directly embed the graph information in our models. We test. We use the first one, we test the second one. So I don't have anything to offer now. We will share, hopefully, if we meet another half a year.
Thank you.
No, I think, yeah, no, it's fine. You mentioned this one. You mentioned that there are early and late fusion methods. What about intermediate ones, where the data is combined from all modalities directly during integration? Have you considered them? Yeah, that's a good point. When I was saying, thanks for the comment, because when I was saying that they are late and early, I don't know if you remember, I also said it's very roughly, very simplistic way to represent that, because they are the intermediate ones that we are testing as well. So we are testing the full spectrum. The intermediate ones will have embeddings that are obtained from a different model and are injected, the embedding, as part of the full model. And then we are exploring with freezing this embedding or continuously updating this embedding. So there are different strategies, and we are testing with all of them because we live in this place where we want it to be as accurate as possible and as efficient as possible, so that when we have new tasks to fine-tune, it will not be a too heavy model. So for these constraints, we are testing different strategies. Thank you very much. Thank you.